Welcome back to DLMP Research and Discovery Seminar Series. After a bit of a break through December, we're back at it, I think every 2 weeks, all the way through the end of the academic year, so through May at least. Some of you may know or have been expecting Dan Landau to be giving the talk today. Due to a scheduling conflict, he was not going to be able to do it. And our current speaker, Marc Vermulst, who was already scheduled, was kind enough to switch to today at short notice. So I really appreciate him doing that. And Dan is going to be giving a talk on April 12th. So if for those people that were hoping to see him, he's still on the schedule, and that's when it'll be. Second, just as a reminder for everybody, especially the students for the Path 520, to make sure you fill out the evaluations at the end of the talk. So without further ado, as I kind of alluded to, our speaker today is Marc Vermulst. I've known Marc for 15 years, and that's because Marc did his graduate work here when it was the pathology department with Larry Loeb. And I met him just at the very end as he was finishing up, and I came in the lab. We overlapped probably about 6 or 9 months. And he was very keen on understanding mitochondrial mutations in the context of aging, and he developed some methods to do that. Was very successful, and then moved on to David Chan's lab, where he continued to work on mitochondrial DNA and looking at protein or mitochondrial quality control, and ended up getting a really nice Cell paper out of that within a very short period of time. It's a pretty influential paper, but he didn't stay there very long, because then he moved to UNC Chapel Hill in the Department of Chemistry, with actually my former graduate advisor, Dr. Dorothy Erie, and where he was working on transcription and transcriptional errors, which is kind of something that I've been very interested in, but I looked at it at a very low level. He took a much broader approach, a high level approach, and looking at it like, what are the consequences of transcriptional errors? And so, as you can see by the talk title today, he's continued on with that work after getting a K99, and I believe he was at UPenn for a couple years, and then he got recruited to USC, where he is an assistant professor, and he's been kind of dabbling in both in transcription error as well as continued work in mitochondrial DNA. With that, I will hand it over to Marc.
Yeah, thank you, Scott. It's wonderful to be here. The only sad part is that I can't be there in person. You already know that some of the best time that I've had in my life has been in Larry Loeb's lab. And a lot of the ideas that I developed there and the things that I learned there, I still apply to this day. The work I'm going to show you today is not directly informed by the experiments that I did there, but they are certainly inspired by them. You know, the type of thinking, the logic, the types of animal models that we use, it's remarkably similar to some of the work that Dr. Loeb has done over many years. What I want to tell you about today is this new idea. Well, it's a recycling of an old idea, perhaps, where we try to look at the impact of transcript errors on human pathology. This idea started off as a very small thing. And it came into being when my lab started looking into the origins of various age-related neurodegenerative diseases, and particularly those that are caused by or associated with the aggregation of proteins. So you all probably know that various diseases like Alzheimer's, frontotemporal dementia, and Parkinson's disease, they're all characterized by the aggregation of various proteins. And in these brain slices you're looking at over here, you see those aggregates stained in brown. What fascinates me, and I think other people as well, is the mechanism by which these protein aggregates form. It seems that they are generated particularly by misfolded proteins. Usually when a protein misfolds, it's not that big of a deal, but in certain rare cases, the misfolded protein can acquire new properties. And the most dangerous of those properties are amyloid and prion-like behavior. The general idea is this. A protein, it doesn't matter what protein it is, misfolds into a particular shape, as depicted by this blue molecule here on the left-hand corner. And if it acquires amyloid properties, it will be able to bind to wild-type proteins of the same amino acid sequence. And then as it binds to them, it will force them to assume its shape. And in doing so, endows that protein with amyloid properties also. So once that binding reaction has happened, this new protein is now capable of recruiting the next wild type protein, and so on and so forth. So in that fashion, through that mechanism, these really long, neatly organized amyloid fibers or various amorphous masses of proteins can potentially form. Because these diseases are endemic in Western society, a lot of people, including me, are very interested in the origins of these initial misfolded amyloid proteins. In some cases, the origins of these proteins are pretty clear. They're caused by genetic mutations, which we know from the analysis of the genome of various patients and their families that carry these genetic changes. And those studies, I think, have been unusually informative. I have personally learned two different things, two main messages that I get out of this. One message is that a single amino acid change can be sufficient in order to greatly increase the propensity for a protein to form an amyloid seed. And the second thing that I think for me was really important was that only a small number of cases of patients that end up in the clinic carry one of these genetic changes. For example, in Alzheimer's disease, I think only approximately 5% of patients that enter the clinic get there as the result of a clearly defined genetic change in a small subset of important genes. The remaining cases, 95% of the cases perhaps, it is not completely clear what caused it, even though these patients carry the same hallmarks as familiar patients do, including nearly identical amyloid plaques and neurofibrillary tangles generated by these misfolded proteins. And what I have been considering, and what other people have thought of in the past as well, is that there is a possibility that perhaps these misfolded proteins, these amyloid proteins, perhaps they could be caused not by genetic changes, as in familiar cases, but through transcript errors, meaning that the gene itself that codes for the appropriate protein, maybe it's APP, maybe it's SOT1, some other protein, encodes for this pre-amyloid protein. It's perfectly fine, but due to a mistake that occurs during transcription, perhaps the exact same mutant proteins can be generated that plague these familial patients. Usually generating a small amount of mutant proteins, even is not that big of a deal. However, these proteins, these amyloid proteins, have this remarkable capacity to quote unquote replicate themselves with the help of wild type proteins, right? So even a small seed, a small amyloid core made of out of proteins that were generated by one of these transcript errors might be sufficient to induce widespread protein aggregation. That's the general idea I'm going to try to convince you of. So this is our general model. We envision that at some rate, which can vary greatly, which I'm going to show you later as well, that transcription errors happen in any gene affecting every transcript. And in some cases, those errors result in proteins with known amyloid or prion-like properties. That those proteins are depicted here on the left-hand side. There is a single amino acid change that is responsible for the amyloid properties of that protein, and then it'll use wild-type proteins in order to grow the seed into these large amyloid fibers that characterize these diseases. That's a general core of the idea that I want to explain to you today. You know, it's if I think about this project at a bigger level, I would probably say that what I'm trying to sell you on is a new mechanism of human disease. Bacteria cause disease, viruses cause disease, genetic changes cause disease. Perhaps in some cases, transcription errors can be responsible for disease also. And perhaps when we don't find the origins of a disease in the genome of the patient, maybe we have been sequencing the wrong thing. I'm not the first to come up with this idea, even though I'm going to show you that we've taken it certainly to the next level and then some. The first inkling that this idea might be true is published in a 1998 science paper that got very little traction. And this was a paper by a Dutch scientist who found that in non-familial Alzheimer's patients, there seem to be transcript errors that happen in the beta amyloid precursor protein that result in small peptides that ultimately become part of the amyloid plaques that characterize the disease. And that was sort of the first clue. There have been others that have proposed similar things. For example, I want to highlight Dr. Prusiner, who got a Nobel Prize for the discovery of prions. And he wrote in one of his reviews in the 1970s, there were 1 or 2 lines only, but there he states that, you know, on the origins of these misfolded proteins, why they misfold in this particular shape, perhaps one of the reasons is that there is some RNA editing problem. So at the time, RNA editing was just discovered, and he envisioned that perhaps some erroneous editing event might happen that changes the code of the RNA, and that results in a mutant misfolded protein that could have these prion-like properties. I am proposing something quite similar. So the reason why it was difficult to follow up on this work and these ideas, the main problem was that there was not a good mechanism at the time to detect these transcript errors. So if you can't find them, how can you study them? So the first task that we had when we come up with this idea and we read about these ideas was to generate a tool by which we can do that. I'm going to show you this tool very briefly. It's not very important, but I want to show you the general thought behind it. And then I want to show you that with this tool, we were able to find transcript errors that we know result in amyloid proteins, that those proteins can then corrupt and convert wild-type proteins to an amyloid state, and that actually very small amounts, the approximate amount that you would get from a transcript error, that number of proteins is sufficient to induce this chain reaction. And I want to show you that that threshold, this ratio of wild-type 2 mutant proteins is routinely breached inside cells for various reasons. And then finally, I want to show you that the cells that make these errors are actually predominantly neurons. So they are cells that are particularly prone to protein misfolding diseases. And then finally, I want to tell you a little bit about the errors that occur at different sites, random errors, and how they contribute to protein aggregation diseases also, and that they might make a good model for recently discovered patients that we found to carry RNA polymerases that seem to be error prone. Okay, so let me start off at the beginning then, and this technique that we developed that allows us to find transcript errors across the human transcriptome. The most obvious way to measure errors is through RNA sequencing. If I imagine getting an extract from cells of various mRNA molecules, some of these molecules will have an error in it, as depicted by this red circle over here. If you process that RNA, you reverse transcribe it, you put adapters on it, and you sequence it, you will ultimately find this error in your sequencing library. The problem, though, is that as you're making the library itself, various artifacts pop up that are indistinguishable from the transcript errors you're trying to find. So, for example, the reverse transcriptases that you use to make cDNA copies are notoriously error prone, and they will introduce base changes that will look just like the transcript errors that you're trying to find. Sequencing itself is very error prone also. So these are all issues that we needed to overcome. And to make this entire project work, what we really needed to do was a way to distinguish between the artifacts that are introduced by library preps and the actual transcript errors present in the RNA templates. That was the real key. There are different ways to do that. Dr. Loeb and his student Kate Bayliss have found one way to do that also. The approach we took is slightly different. Our technique relies on the circularization of RNA templates. So prior to reverse transcription, the only thing that we do different is we circularize the RNA molecules prior to reverse transcription. And by having these RNA circles, we're able to reverse transcribe them in a rolling circle fashion, meaning that reverse transcriptase can go around this RNA template multiple times, and each time it goes around the circle, it'll add one copy of the template to this growing cDNA molecule. And if there was an error present in the template, then that error would be present in each of those copies. In this particular case, in the cartoon that I'm showing you here, the reverse transcriptase went around the circle 4 different times. So there are 4 copies of the same template in tandem in this single cDNA molecule. And then through bioinformatic means, we're fishing out the differences with the genetic background of this cell that are present in each of the copies. And an artifact that's introduced by sequencing or reverse transcription will occur in a semi-random fashion, and thus will only be present in 1 or 2 copies of this tandem repeat cDNA molecule. So that is the general gist of it. It's that's the technique that we've been using. We've let it loose on many animal models. We've sequenced yeast and worms and flies and mice and human cells now also. In this data set that I'm showing you here, which consists of about 280,000 transcript errors that we found, will be published in PNAS later this month. Find everything that you can possibly think of: insertions, deletions, base substitutions. But from this data set, this 280,000 errors, what we really wanted to do is search for those that may result or are highly likely to result in amyloid proteins. So that was one of the first tests that we did. We basically cross-referenced our error database with mutations that have been found in patients with amyloid diseases. We found a whole bunch of them. In this particular case, we found a transcript error, TE in blue over here, where a guanine has been replaced with an adenine, generating a protein that has also been found in a family of which multiple family members suffer from amyotrophic lateral sclerosis. So we thought that this error might replicate that protein that's causing the disease. To test that idea, we first expressed both the wild type version and the version that would be generated by the transcript error in human cells. On the left-hand side over here, you see the expression of the wild type protein tagged with GFP. You'll notice that the distribution of the protein is very equal across cells and different compartments. And if you compare that to the mutant version generated by the transcript error, you'll see that in fact the protein is forming these speckled protein aggregates throughout the entire cell. You'll also notice that the nucleus is completely devoid of the SOD1 protein. Both of these things, the fact that the protein aggregates into large clumps and that it is excluded from the nucleus, are both phenotypes that you see in ALS patients also. If you look at the proteins a little bit deeper and you use transmission electron microscopy, you'll see that this protein generated by the transcript error does indeed result in these amyloid fibers that typify various amyloid diseases, while the wild type does not. The most important thing, though, if this idea I'm telling you about is true, is that not only should the protein generated by the transcript error result in protein aggregation, but it should also be able to convert and corrupt the wild type proteins, right? And in like that way, grow into these large amyloid fibers from these small cores. And that is what's being tested here partially. We're expressing again the version of SOD1 that was generated by the transcript error, tagged with M-cherry. You see some large protein deposits inside the cells and exclusion from the nucleus. And now we express in the very same cell also the wild type version. And you can see that instead of being equally distributed throughout the cell in both the nucleus and the cytoplasm, now the wild type version forms aggregates with the mutant version also. So they are together in the same protein aggregates, suggesting that the wild type version is indeed being corrupted and converted into an amyloid version by the molecules generated from the transcript error. But that's, I guess, just a quick catch up. So what I've shown you up to this point is that we've optimized this new technique to detect transcript errors. And we found indeed errors that result in known amyloid proteins, and that these proteins seem to be able to convert wild type proteins to an amyloid state. What is really important, though, is that we understand how many of these mutant proteins, how many mutant transcripts are needed in order to initiate this entire process, because transcript errors are still rare, at least most people think so, and that can be debated, as I'm going to show you later. And so for this idea to be interesting, a small amount of transcript errors should be sufficient to initiate this. I'm going to show you that data with the help of another protein, p53. One thing that we considered when we were looking at our database was that in addition to the errors that result in known amyloid proteins, there might be lots and lots of errors in this database that result in amyloid proteins that haven't been described in the literature yet. So we collaborated for that purpose with Dr. Lin Chen and his postdoc Yi Ku, who work in the molecular and cellular biology department across the street. They're very interested in carcinogenesis. And we had a database of errors that specifically affected the TP53 protein, one of the major guardians of the human genome, and a protein that is mutated in 50% of human cancers. Dr. Chen and Yi wrote several algorithms to try to identify errors that might result in amyloid versions of TP53. It's actually known that about 15% of human cancers have an TP53 that displays amyloid and prion-like behavior. And he found among those errors one that he predicted to result in an amyloid version of this protein, which he named S149F for the amino acid change that it caused. Dr. Chen is a biophysicist, so he used various biophysical and biochemical tools to investigate these wild-type and mutant proteins. One technique he used is dynamic light scattering. It's a technique by which you can measure the radius of protein particles. So Dr. Chen expressed wild-type p53 and mutant p53 in insect cells and in bacteria, and then put them under this dynamic light scattering technique to see how large the protein particles were. When he examined wild-type p53, he found that most of the molecules in the solution had a radius of about 50 nanometers, which is approximately the size of a monomer of p53. And then when he examined his mutant version, he found that most of them had a radius that was much, much larger, indicating that tens or even hundreds of p53 proteins had aggregated together into these large clumps. So that was already really exciting, that we were able to predict new versions of amyloid proteins. And Dr. Chen had something even more interesting then, when he took 1% of this mutant TP53 solution and dunked it into the solution that contains the wild-type p53. And as a result of adding this tiny amount of p53 protein, he was able to show that there was a disproportionate increase in the p53 aggregates of very large sizes. He examined the same conditions with other tools also. So for example, you see again transmission electron microscopy images here. On the left-hand side, wild-type p53. On the right-hand side, the mutant version generated by the transcript error, which forms these large amorphous aggregates. And then in the middle is a solution where 1% of the error-prone p53 was added to the wild-type solution, and many small protein aggregates formed as a result. You can also examine those aggregates with atomic force microscopy, a technique that Scott will be very familiar with. And you can see that there are these sort of fibrous aggregates formed as a result. You can try to grow crystals also, and you can see with your naked eye, you can see in those crystals the formation of these aggregates. If you add it's a little bit of p53, and even if you look at an Eppendorf tube and you add a bit of mutant version of p53 to the wild type version, you can see eventually these large aggregates, these gel-like blobs floating around in the solution. Okay, so not only do transcription errors result in amyloid proteins, they can convert them to an amyloid state, and very small amounts, 1%, seems to be sufficient to cause this protein aggregation phenomenon. Now, is that 1% threshold, which is arbitrary at this moment? Dr. Chen is currently diluting these conversions down to smaller numbers and doing the same experiment. But we can already ask, is this 1% threshold, 1% wild type versus mutant protein, is that breached inside cells? Here, again, I was able to use the knowledge that I gained from working in Dr. Loeb's lab. One of the major themes throughout his research has been that proteins are fallible. That's the main message that I got out of Dr. Loeb's lab. Proteins are not that good, or not flawless, at least at what they do, and there are several mechanisms by which you can make these proteins more error-prone than they already are. Dr. Loeb primarily works on DNA replication, and it is very well known from his work and work of others that if a template strand, a DNA template strand, has been damaged, in this case, we've introduced an O6-methylguanine lesion, then DNA polymerases can have trouble putting the correct base opposite this damaged base, because the damage warps the base pairing properties. If DNA polymerases make these errors, it is pretty reasonable to think that RNA polymerases will make similar errors also, because RNA polymerases depend on the same base pairing properties in order to facilitate their fidelity. So what we were thinking was that if we damage cells with a compound that introduces these O6-methylguanine lesions, and we allow the cell to then transcribe these damaged genes, in this case, as an example, I'm using the RPS16 gene, there is a chance that the RNA polymerase will put in the right base or the wrong base. And we wanted to know what that ratio is. Does it cross this 1% threshold? So that's exactly what we did. We used single cell sequencing technology coupled with custom-made bioinformatic pipelines to do the analysis. And for RPS16, we found that in one single cell, 91 transcripts were detected. 53 of those were wild type, and 38 carried an identical change in the transcript, and it was the change that would be expected from an O6-methylguanine lesion. It is, and that was not visible in any of the other cells. It is important for me to tell you as well about this experiment that they were done on arrested cells. So if cells are arrested and are not replicating their genome, there is no way for DNA damage to be fixed into a mutation. So there is no way that these results that I'm showing you are the result or confounded by DNA mutations. The general message, though, is that if you damage cells with a chemical compound that introduces certain lesions, you will get an enormous amount of erroneous transcripts generated throughout the entire genome that are at levels of up to 80%. So somewhere between 5 and 80% of the transcript made from a damaged gene will carry the same identical change, vast, easily crossing this 1% threshold that Dr. Chen has been experimenting with. The role of 6-methylguanine is getting greater interest now in the context of age-related neurodegenerative diseases because of a recent finding. You may or may not know that a genome-wide association study had found that loss of or down regulation of MGMT, which is a DNA repair gene that repairs 6-methylguanine lesions, has been found with an increased progression and severity of the disease. So together with Dr. Caleb Finch and his postdoc Max Torwart, we replicated that result, and we found that in normal patients with 2 copies of the ApoE3 allele, MGMT levels were normal. But if you introduce one ApoE4 allele, which is gives you a predisposition to Alzheimer's disease, or you look at Alzheimer's patients that have two ApoE3 alleles, and various combinations of these work as well, there is a significant downregulation of MGMT expression, meaning that if DNA damage is created, specifically that damage that creates transcript errors, that damage will remain for extended periods of time. We mimic that in various experiments. So for example, here, again, we're treating cells with MMNG. That results in a big increase in transcript errors. And then over time, 2 hours later, 6 hours later, the error rate goes back down as the cell is repairing its genome and removing the very lesions that cause the errors. But if you do that experiment in cells that are repair incompetent, because we have removed the DNA repair gene that's the homologue of MGMT, you can see that the error rate remains very high for extended periods of time. So we think that that is one of the reasons that loss of this DNA repair gene is associated with accelerated progression of the disease. There are other mechanisms by which you can increase the error rate also. For example, in aging yeast cells or aging flies, you see an increase in the error rate of transcription with age. Specifically in aging animals, there's an increase in these C2U changes, which happen to be the exact changes caused by 6-methylguanine. That's not to say that that's what they're actually caused by, but they happen to be the same ones. There are various genetic sequences as well that make a gene more error prone. So for example, in this paper that I quoted you earlier, where Dr. Van Leeuwen, a Dutch scientist, found that there are transcription errors occurring in the APP gene, those events seem to occur at repeats of GA dinucleotides. And when we examined our database of transcript errors and we looked at the error rate of dinucleotide repeat sequences versus any other part of the transcriptome, we found that in each case, the error rate of transcription goes up with increasing length and with certain compositions of the dinucleotides, up to 100-fold if the dinucleotide is consists of 2 identical bases, and up to 12-fold in the case of disparate bases. So ultimately then, transcript errors happen everywhere across the genome. They can create these amyloid proteins. Very little amyloid protein or transcript errors are needed in order to generate these harmful amyloid proteins that's regularly breached inside cells, and certain genes, including genes that are essential that are important components of the pathophysiology of Alzheimer's disease, have sequences in them that make them more error prone. We are also considering, what cells are making these errors the most. If these errors happen in intestinal cells only, maybe we wouldn't care as much. But there is a possibility that certain cells make these errors more frequently than others. So in order to investigate that, we made use of a new mouse model, which we received from Dr. Jeffrey Strathern. For the purposes of timing, I want to not explain the model in detail, but suffice it to say that it carries a mononucleotide tract, so a repeat sequence that is relatively slippery. And if an error occurs in that sequence, it results in a permanent change elsewhere in the genome that leads to permanent YFP expression. So a temporary event, this transcript error in what happens to be the CRE gene, results in a permanent change inside cells in the genome of the cells that can be that results in permanent YFP expression. So that means that we can now slice the tissues of these animals into very small pieces and look for cells that express YFP. And those are the cells that have undergone at one point in their life span a transcription error event. When we look at different tissues, intestine, liver, testes, all kinds of other tissues, we find very few cells that are YFP positive. But if we look at the brain, and particularly at the hippocampus, there are many regions that are filled with these YFP positive cells. Some of you might know that in the hippocampus of the brain, there are various groups of neurons, CA1 neurons, CA2, CA3 neurons, and dentate gyrus neurons are among them. And CA1 neurons are particularly sensitive to Alzheimer's disease. And as it turns out, those are also the cells that carry the most YFP positive cells. Oops. And the dentate gyrus is a little bit less affected compared to the CA1 neurons, and they have an appropriate lesser amount of these YFP positive cells also. So not only are the genes that play a really important role in Alzheimer's disease have these error-prone sequences in them, the cells that are affected by the disease or that mediate the disease are very error-prone on their own. Okay, the final thing I want to tell you about is random mutagenesis. So I've shown you a lot of data today about transcription errors that occur at very specific spots, right? Those errors are very rare. There will be many errors, though, that occur elsewhere in the transcriptome that may not affect a very important component of a protein at all. What is the impact of those errors? And could they contribute to loss of proteostasis also? To investigate that idea, we used error-prone yeast cells. We've now made error-prone human cells also, but that data is not finalized yet. Error-prone yeast cells that display an increased error rate of transcription. So, for example, we found cells that have lost a gene called RBB9 or have a mutation in a gene called RBB1 that display about a 5-fold increase in transcription errors compared to wild-type cells. And we simply investigated the health of the wild-type cells to the error-prone cells. This slide I'm showing you here is one small component of the data set that we generated. We did a proteomics screen, and we looked for proteins that were significantly upregulated in the error-prone cells compared to the wild-type cells. We found 17 proteins and above a certain threshold also. And of the 17 proteins that we identified, half of them were molecular chaperones. So these are molecules, heat shock proteins and various other molecules whose sole purpose it is to refold misfolded proteins. So apparently, these error-prone cells suffer from a loss in proteostasis and an excess amount of misfolded proteins that is now being addressed by the upregulation of these misfolded, of these molecular chaperones. And that makes perfect sense because if you replace randomly in a protein a hydrophobic amino acid for a hydrophilic one, a polar for a non-polar, etc., the most likely result is not that the protein gets a new function. The most likely result is that the protein will misfold. And that has been really elegantly shown in another one of Dr. Loeb's papers, together with his grad student, Highway Go. Might these random proteins contribute then to disease states in humans also? This is the general idea that we're working with. We're imagining that toxic proteins are generated through different means, transcript errors being one of those means. Usually, those proteins are rapidly degraded by the protein quality control machinery, proteasome, autophagy, etc. If you simultaneously, though, also generate all these non-toxic but still misfolded proteins, you will clog up the protein quality control machinery, allowing these toxic proteins to evade degradation longer than normal and allow them to form seeds and to generate these protein aggregates. So it's really creating this traffic jam of misfolded proteins in front of the protein quality control machinery, that even though these proteins are not toxic on their own, they still delay the degradation of proteins that are toxic. We generated a lot of data for that as well. You can read about it in our Nature Communications paper from 2015 if you're interested. But in essence, what we did is we expressed various toxic aggregation-prone proteins in wild-type and mutant yeast cells, the wild-type and the error-prone versions. And in the error-prone cells, there were greater and more protein aggregates generated from TDP43, A-beta, Huntington, prions, etc., in the error-prone cells compared to the wild-type cells. There are various reasons by which we think this is caused by these traffic jams in, for example, the proteasome. I can't show you all that data today, but it's all described in this paper. And then I think that some of these models might be more useful for not just basic biology, but also for clinical diseases. I recently got in contact with some folks from Holland who have described pediatric patients that carry mutations in a gene called POLR2A, which is the major catalytic subunit of RNA polymerase II in humans. Some of the mutations described in these patients are right next to locations that are essential for the fidelity of RNA polymerase II. So, for example, there is a location in this protein, amino acid 1249, where if you mutate that allele, it disables the connection between POLR2A and a fidelity factor. And one of the patients carried a mutation at amino acid 1251. We have generated cells that are error prone as a result of a mutation in amino acid 1126, and two of the patients have a mutation in amino acid 1124 and 1125. So we were very curious to know whether or not these mutations result in error-prone RNA polymerase IIs, and we found through both experiments in yeast and in human cells that is indeed the case. So there are, in fact, human patients that carry error-prone RNA polymerases. So that's the end of my talk. Thank you so much for listening. I hope I've shown you, something relatively new, a simple idea that the proteins that cause amyloid diseases do not have to be generated by genetic changes. They can be generated by transcription errors also, because the proteins themselves are have the ability to replicate themselves with the help of wild-type proteins. It's becoming clear that these amyloid proteins are ubiquitous throughout the human body as a function of age. It is responsible for heart disease, can contribute to cancer, in addition to these neurodegenerative disorders. So I think that maybe this idea can be transferred not just to various neurodegenerative diseases, but also diseases of other tissues. I hope that I've shown you that they indeed give rise to known and unknown aggregation-prone proteins, that they can convert these wild-type proteins to an amyloid state, that DNA damage, a super common feature, maybe the main feature of aging organisms, that is a super powerful driving force behind transcriptional mutagenesis, and can in fact generate many transcripts that carry an identical change. But the error rate itself is not stable. It changes as a function of age and various other features too. There is a Nature Communications paper coming out next month from us, and a PNAS paper that describe the various components of the genome and the epigenome that control the error rate. If you're interested, please check them out. There are certain repeat tracks that are present in essential genes for various diseases that make them more error prone, and that there are cell types, specifically cells that are involved in these diseases, that are more error prone than other cells. And so the final conclusion that I would like you to think about is that potentially familial and non-familial cases of protein misfolding diseases are caused by the same mutant proteins. It's just that the way they are generated is different. In familial cases, it's a genetic change. In non-familial cases, potentially they are mutations that occur at the transcript level. The work that I showed you was performed mainly by 4 different people. Sarah Shemtov, she did a lot of the protein aggregation work and the sequencing efforts. Marc worked with the mouse model that I showed you that we received from Dr. Strathern. Claire is a sequencing expert and truly amazing at it. Dr. Gout has, who works at the University of Mississippi, has done our bioinformatics. We're extremely grateful for the help of Dr. Chen and Dr. Koo and the mouse model from Dr. Stratton, and received support from other lab members also, including Ronald Tony, Andrew Uridian, and Alex Park. And I just want to quickly mention that we are looking actively for new postdoctoral scholars. So if there are grad students out there or postdocs looking for new jobs, please send me an email. Thank you. I'll take any questions you might have.
Thanks again, Marc. That was a great, super interesting talk. So I'll just open it up for questions right away. Anybody?
Hi, Marc. I can ask a couple questions. This is Keith.
Oh, hi, Keith.
Anyway, so one of the questions I had is when you were looking at that 1% abnormal converting the wild type to aggregates, was that done in solution or is that done while it's being co-expressed?
Oh, sorry. Yeah, you're right. That's done in solution.
So in solution, and you don't need to unfold them at all to get that to aggregate?
No, I think what Dr. Chen did, he took 1% of the mutant protein aggregates, but he fractionated them in such a way that they were approximately a certain size, 60 to 100 proteins. And the reason for that is that 60 to 100 proteins is what you might expect from a single transcript error.
Okay, and then you were saying that the same proteins would cause the aggregation. How about if you had different proteins, but they bound together? Can one mutant protein that causes this aggregation cause totally different protein to aggregate?
I think it has to be pretty much the exact same amino acid sequence. They can form different types of aggregates though. If you look under various cryo-EM techniques at these protein aggregates derived from patients, you'll find that with the exact same mutations, you'll find different forms of these aggregates, and those correspond to different phenotypes in the patients. So one thing we're trying to do is we're trying to use cryo-EM to see whether or not the aggregates made by the transcript errors have the same structure as those from known patients.
Anybody else?
Yeah.
I have a question.
Oh yeah, go ahead, Martin. Larry, you'll be next.
Yeah, my question is about what is known actually about the human brain regions that are mostly involved, that are the usual suspects in neurodegenerative diseases. We know that pathologies unfold in specific anatomical regions for Alzheimer's disease, for Parkinson's disease, for other forms of dementia in different patterns. What are your thoughts on whether or not these could be also hotspots for these transcriptional errors that are performed, or perhaps even changes in RNA polymerases in these regions? Have you looked at that? It seems like your PNAS paper is mostly on embryonic stem cells, but so far there is no other real evidence in the human brain that these hotspots align with your hypotheses. I mean, the mouse data are really.
I'm speaking now. I'm supposed to speak next, and I don't have to.
What?
Yeah, so let me answer that. We're sectioning the brain in a gazillion pieces, and we're walking through the entire thing to find hot spots and groups of cells that are particularly sensitive to transcriptional mutagenesis. One region that we have identified so far is the Purkinje cells in the cerebellum. You may or may not know that those cells are particularly involved in prion diseases. We're homing in on the substantia nigra now. That's one of the main targets that I'm the most interested in because of the increased amount of oxidative damage generated by those cells, and that could lead to increased transcriptional mutagenesis through the mechanisms that I showed you earlier. But we don't have that data yet. Secondly, we haven't worked with human brain samples yet. What we have done, though, is that from the human embryonic stem cells, one of the reasons why we picked those cells, one reason is that they've been described in great detail by the Human ENCODE project. So all the molecular details are known in great detail, so we can correlate the errors we find with specific features of those cells, but also because we can induce them to become different cell types. So we have generated human neurons from the H1 ESCs that we've been using, and we are currently in the process of sequencing that data. And I hope to give you an answer to that question.
Very interesting. Thank you.
All right, Larry, last question.
Okay. I have three little questions. First, do we know definitely that there's no RNA replicase in human cells?
But there is some mechanism by which RNA molecules are replicated on their own in the absence of DNA directed. I actually don't know the answer to that. I don't know of any evidence, but that doesn't mean much.
What's the story with respect to the thought patterns of using known purine analogs that have been tested in cancer and measuring their effect on the progression of Alzheimer's disease?
Right. So I'm extremely interested in chemotherapeutic agents. So a lot of these chemotherapeutic agents generate alkylating DNA damage. In our hands, that's the most powerful source for transcriptional mutagenesis. So we're testing out temozolomide and various other drugs. What is certainly clear is that tissues, you already know this, tissues that have been exposed to these chemotherapeutic agents or certain radiation treatments, they age at an accelerated pace, right? Your tissues are 20 years older after treatment compared to before at a biological level. I think, I don't know the exact reason for it. It probably has to do with cellular senescence and various other things, but I am extremely curious to know whether or not the exact chemicals that you're talking about right now, what impact they have on the fidelity of transcription. I'd like to mention in this context also paraquat and rotenone. So rotenone is a chemical that is used to make Parkinson's mice, right? And they work by disturbing mitochondrial function and generating enormous amounts of reactive oxygen species. Paraquat has a very similar role. Farmers that use these chemicals, which in some ways may mimic to a certain degree the chemicals that you're talking about, have a greater incidence of Alzheimer's and Parkinson's disease. I have been asking around the exact same question that you just posed. I haven't gotten a clear answer yet, though.
Did it, Larry?
Yep.
Okay. All right. Well, we are over time, so I want to be respectful of everybody's time as well as Mark's. So thank you again for giving the talk, especially switching at the last moment. And,
No problem. Thank you so much.
Super interested in as this moves forward. So thanks again, and see you in a couple of weeks.
All right. Bye, guys.